Okay, in this portion of the Chapter 3 discussion, we're going to be dealing with the organelles and the components inside the cell in a little bit more detail. The cytoplasm is simply the material that's contained between the cell membrane and the nucleus of the cell, and it's filled with organelles, cytoskeletal elements, waste products, nutrients, electrolytes, and water. Okay. Um, the material that's suspended in the cytoplasm isn't necessarily stationary. It can move about. Some of this movement is passive, and some of this movement is active, meaning it involves the use of ATP. In addition to organelles, occasionally you'll see inclusions in these cells, and these may be collections of molecules of nutrient content, they may be collections of pigment, and they may be collections of certain waste products. It all depends on the cell type. So you could think of an inclusion as sort of a closet where certain things are stored in the cell. The organelles inside the cell fall into one of two categories, either membranous or non-membranous. The membranous organelles have a membrane similar to the plasma membrane surrounding them, and the non-membranous organelles do not. The purpose of the organelles is to allow chemical reactions that sustain life to take place in a small enough space that they are efficient. And it also separates chemical reactions that would otherwise interfere with each other so that they don't. Okay? So we're going to look at in detail each of these organelles and describe what they do. And the first one is the mitochondria. The mitochondria is made up of a double membrane. There's an outer and there's an inner membrane. Let's use laser pointer to highlight. Okay. The outer membrane is similar in composition to the plasma membrane, and the inner membrane is similar in composition to bacterial membranes and is folded into ridges and grooves that are called cristae. And the purpose of these folds is to increase surface area so that we can carry out metabolic reactions. Underneath the inner membrane, in the core of the mitochondria, is what's called the matrix, and the matrix contains ribosomes that are similar to bacterial ribosomes and DNA similar to bacterial DNA. And the chemical reactions that go on in the matrix are also part of the collection of chemical reactions that generates ATP for the cell, and those are the Krebs cycle reactions, right? The electron transport reactions happen in the inner mitochondrial membrane, right? And the buildup of protons from the electron transport chain that ultimately drives the production of most of the ATP happens in the inner membrane space between the outer and the inner membrane. Okay? These are thought to have descended from ancient bacteria, and the reason for that is the presence of mitochondrial ribosomes that are more similar to bacterial ribosomes than eukaryotic ribosomes, and also the sequence of mitochondrial DNA mimics that of bacterial DNA. And these organelles also replicate on their own schedule according to the cell's need for energy. Okay? In order for them to produce the maximum amount of energy for the cell, they require oxygen. They generate 20 times more ATP in the presence of oxygen than without it. The ribosomes are the site of protein synthesis, and these are non-membranous organelles that have two subunits, a large and a small, and they can be found either attached to the rough endoplasmic reticulum, where they generate proteins through a process called translation that ultimately enter the rough ER and fold into their native conformation, are then loaded into a transport vesicle, which then takes them to an organelle called the Golgi Apparatus, which then loads them into a secretory vesicle, which takes them either to another organelle or to the plasma membrane, where they can either become part of those organelles or membranes, or in the case of fusing with the plasma membrane, they may be exocytosed, ejected into the outside environment. The free ribosomes manufacture proteins that stay within the cytoplasm, and many of these are enzymes. The endoplasmic reticulum is simply a large maze of connected membranous sacs that in one region called the rough ER collects the translated proteins that will ultimately become either embedded into or extruded from a membrane target, and the smooth ER, which is the site of lipid biosynthesis and also calcium storage and the breakdown of toxins inside the cell, and it lacks ribosomes on its surface. So the rough ER synthesizes membrane integral proteins and phospholipids, which then move to the ER interior, get enclosed in a transport vesicle, and then get moved to the Golgi, while the smooth ER, which is continuous with the rough ER, has enzymes that are involved in lipid and sterol metabolism, cholesterol metabolism, hormone synthesis, and making the lipids that go into structures called lipoproteins, which are used to move lipid around in the blood. They're also important in the absorption, synthesis, and transport of fats, and the detoxification of drugs, pesticides, and carcinogens. They also convert glycogen into free glucose for use in metabolism, and they also serve as an area to store and release calcium. The Golgi apparatus are stacked and flattened membranous sacs that modify, concentrate, and package protein and lipid from the rough ER. Transport vesicles from the ER fuse with the convex cis face. The proteins get modified, tagged for delivery, sorted, and packaged in vesicles. The three types of vesicles that bud from the concave transface include secretory vesicles, which go to the transface and release export proteins by exocytosis, vesicles of lipids and transmembrane proteins destined for plasma membrane or organelles, and lysosome containing digestive enzymes that ultimately get delivered to the lysosome or become a new lysosome. Okay? And these are extremely destructive enzymes, so they have to be kept inside the lysosome or else they would destroy the entire cell. Peroxisomes are membranous organelles that are designed to destroy free radicals, which tend to break down healthy molecules and produce damage that can ultimately lead, in some case, to the production of cancer cells. They detoxify harmful substances, they synthesize fatty acids, they neutralize these free radicals with oxidases that can convert hydrogen peroxide into water and oxygen, right? The oxygen can be used in the production of ATP, and the water, of course, becomes part of either the intracellular or the extracellular fluid. The lysosomes are essentially the stomach of the cell, and they contain powerful proteolytic enzymes that can break down almost anything that's delivered to them. They destroy ingested bacteria and toxins, and they break down organelles that no longer work. They also break down and release glycogen, and ultimately, when the cell can no longer function, they engage in a process called autolysis, in which they destroy the cell from the inside out so that it can be replaced by new cells. They also break down bone and release calcium in their action inside cells that are called osteoclasts. So let's take a look now at the endomembrane system.
A secretory protein is synthesized inside the rough endoplasmic reticulum, migrates through it, and exits inside a vesicle. The vesicle travels through the cytoplasm to the Golgi apparatus. The Golgi apparatus modifies the protein. The protein is dispatched from the Golgi in a transport vesicle that then fuses with the plasma membrane, releasing the protein to the outside of the cell.
The cytoskeleton has a variety of roles. It's important in maintaining the shape of the cell, but it also serves as a way to anchor cells to either extracellular matrix or to each other, and also as a method for moving organelles and chromosomes around inside the cell using ATP as a power source so that they can do their job effectively. The three types of cytoskeletal elements include microfilaments, intermediate filaments, and microtubules. Microfilaments are the thinnest of cytoskeletal elements, and they're made up of actin. Each cell has a unique arrangement of these strands. Dense webs of microfilaments are attached to the cytoplasmic side of the membrane in what's called the terminal web, and that gives the cell strength, compression, and resistance. They're important in cell motility, in cell shape change, such as in amoeboid movement, where the cell can crawl across a surface from one location to another, and in the process of endocytosis and exocytosis. The intermediate filaments are made up of protein tetramer fibrils that resist pulling force on the cell and attach to cell junctions called desmosomes and hemidesmosomes. And we see these as neurofilaments in nerve cells, as keratin filaments in epithelial cells, and as a variety of supporting structures in other cell types. Okay, so you could think of this as sort of a framework cytoskeletal element. The microtubules, on the other hand, are the largest of the cytoskeletal elements. They are hollow tubes that radiate from something called the centrosome, and they're made of protein subunits called tubulin. They generate these hollow tubes from subunits of tubulin that are attached to each other, so they form a quaternary structure here. And they can determine the shape of the cell, but also the distribution of organelles, and they're very active in a process known as mitosis and meiosis, where they are utilized to move duplicated chromosomes from one end of the cell to the other in preparation for cell division. In mitosis, remember, the cell division produces two cells that have the same amount of genetic material, whereas in meiosis they generate daughter cells with half the amount of genetic material in the original cell. These motor proteins that reside in these transport and secretory vesicles interact with microtubule components using ATP for power, and that allows these vesicles to get to their ultimate destination. So you could think of these microtubules in a sense as train tracks almost, and the vesicles as cars on the train, and the little motor proteins as the locomotive, right, and the wheels of the train. Okay? So again, all of this is active transport when you're talking about involving the microtubules. Now the centrosome and the centrioles are involved in the manufacture and the breakdown of microtubules. So if you think of the microtubule as the line in a rod and reel, you could think of the centriole and the centrosome as the reel of the rod and reel, right? And then whatever the microtubule attaches to would be like the hook attaching to the fish, okay? And what happens then is that they are able to break down or manufacture microtubule components when and where needed, and that helps to control the composition of the cytoskeleton, but also in cell division to guide the movement of the chromosomes to opposite ends of the cell. And of course, all of this synthesis involves the use of ATP for power. So let's take a look at cilia and flagella.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella, or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
So, when we're talking about cilia and flagella, we're talking about movable extensions on the surface of cells. When we're referring to cilia, these are usually apical extensions used to move material across the apical surface from one location to another using ATP for power, and you can see what the power stroke looks like here. Whereas flagella are designed to propel a single cell from one location to another, and the only cell in the human body that has a flagella is the sperm cell, and that's important because the sperm cell has a long journey to get from the tip of the penis all the way to the ovulated egg in the female reproductive tract and faces many obstacles along the way. So at the very least, it has to have some method of propelling it this great distance. Other cellular extensions include microvilli. These are finger-like extensions of the plasma membrane with an actin core, and they don't move, but their purpose is to increase surface area used for secretion and absorption. So a typical location for microvilli would be along the epithelial tissue that lines the digestive system. Okay, epithelial tissue, we're going to find out, is a tissue type that is capable of forming barriers and doing secretion and absorption because of the nature of the cell shapes and the cell junctions, as well as the nature of their cell surfaces. Now, the nucleus is the largest organelle, and it contains chromatin, which is a complex of DNA, which we know as the genetic material, combined with protein, and the proteins in eukaryotic chromatin are both histone and non-histone proteins. Okay, we're going to find out that the histone proteins are there primarily to protect and compress the information so it's not as easily damaged, and the non-histone proteins are primarily used to regulate when transcription happens. Okay. The nucleus responds to signals, and it dictates the kinds and amounts of proteins manufactured in the cell. Remember that the proteins are the tools of life. Most of our cells are uninucleate, with the exception of osteoclasts, and you could argue for megakaryocytes in a way. The rest are either anucleate, meaning no nucleus, such as in platelets and red blood cells, or have a single nucleus. The purpose of the nucleus is to store the chromatin, but also to allow a method for materials to get in and out of the region where the chromatin is located, even if they are of a significant size. And that's one of the reasons that the nuclear membrane, as you can see in this drawing, has numerous pores around it in order to permit the movement of material into and out of the plasma membrane, primarily through passive transport, in addition to the fact that it also has channels and pumps for other types of active and passive transport as well. Okay? And you can see that the outer nuclear membrane is continuous with the rough endoplasmic reticulum as well. Now, the nuclear envelope is a double-layered phospholipid bilayer. So you've got an outer phospholipid bilayer continuous with the rough ER, and then a space, and then an inner phospholipid bilayer that surrounds and is attached to the chromatin. All right? And of course, the nuclear pores span both of these sets of membranes. The inner lining helps to maintain the shape of the nucleus and serves as a scaffold to organize the DNA as a result of interaction between nuclear protein and inner nuclear membrane protein. And the pores, of course, allow the movement of large molecules or complexes into and out of the nucleus. And that's important because The nucleus is the place where ribosomes are assembled and where messenger RNA is manufactured. And those are of considerable size, and they have to be able to get in and out of the membrane in order to do their job, in addition to the fact that proteins that control transcription also have to get access to the nucleus and to the chromatin as well. The nucleoli are areas inside the nucleus that usually stain dark with a nuclear stain because they are the site of the synthesis of ribosomal RNA, which is a structural component of the ribosome. So you could think of the ribosomal RNA as sort of the cement that binds the ribosomal proteins together so they can form a large and a small subunit. The nucleolar organizing regions are attached to the nucleoli, and these are simply the ribosomal RNA genes which are transcribed to make the ribosomal RNA. And there's usually between 1 or 2 per cell. They're more prominent in cells that more extensively utilize protein. The chromatin, as we discussed, is a complex of double-stranded DNA and histone proteins which are used to compress and protect the information so that it's not as easily damaged. They're arranged in units called nucleosomes, which pack the DNA that is involved in gene regulation. These ultimately, during cell division, condense into chromosomes, which are visible, compressed regions of chromatin that contain the genetic information necessary to run the cell. Okay, so prior to mitosis, as we're going to find out, you'll have compression of the chromatin, and the duplicated chromosomes will become visible for the first time. That usually happens during prophase, and then we'll have attachment of microtubules to the constricted region in the center of the duplicated chromosome, which is called the centromere. And then by applying force to either end of the centromere, we'll be able to pull apart the duplicated chromosome and drag one molecule each to opposite ends of the cell. The result is that each new cell will have a complete complement of chromosomal DNA utilized for it to carry out the work of making the tools of life. Okay, so nobody's missing any information. And normally this copying is 100% accurate, but occasionally mistakes are made, and mistakes are called mutations. And mutations can have one of 3 consequences. They can be beneficial, meaning they will produce proteins that generate characteristics that make it more likely that the organism will be able to live long enough to breed. They could be detrimental, which means they won't. They'll make it less likely that the organism will live long enough to breed, and they may not have any effect at all. Okay? And we'll see in our discussion of mitosis and meiosis exactly how this occurs. Okay? So I'll join you in the next segment.